Welcome back to Outbreak News This Week, your source for all the news about worms and germs. Here's your host, microbiologist and editor of OutbreakNewsToday.com, Robert Harriman.
Well, welcome back to the program. Now, Clostridium difficile, or C-diff, as many of you know, and the topic of fecal microbiota transplantation, or FMT, has been a topic on the show several times. And this evening, I'm joined by the senior author of a study published in the journal Alimentary Pharmacology and Therapeutics. And what they do is they compare the use of fresh fecal material with frozen and freeze-dried products for the FMT. Welcome to the show, Herbert DuPont, MD. Dr. DuPont is a professor in the Department of Epidemiology, Human Genetics, and Environmental Sciences at UTHealth School of Public Health. And he is also the director of the Center for Infectious Diseases at UTHealth School of Public Health. Dr. DuPont, sir, welcome to the show.
Thank you so much.
Yes, sir. I'm so glad you could come. I enjoyed reading your study, and I want to talk about your research. But before we do, just as a reminder for my audience, I just want a little primer for them. Can you explain what is C. diff and what is C. diff disease?
Well, C. diff is an intestinal infection, which is now the number one most important cause of hospital-acquired infection in the United States. It's more important than methicillin-resistant staph and other infections that are acquired in the hospital. It's number one. And to give you an idea how important it is, the CDC estimates that there are 29,000 deaths in the United States every year from C. diff. People, unless they've had it or had a family member with it, have never heard of it.
Right.
But it's one of those silent diseases that wreaks havoc and kills people. And one of the challenging problems of C. diff is that it's a disease that recurs. And when it recurs, and people can't get well from it without one of these transplants.
So what is a fecal microbiota transplant?
Well, first of all, there's a whole lot of diseases that are associated with abnormal intestinal microbiota. We have in our GI tract 100 trillion bacteria, which is 10 times the number of cells of the body, 200 times more DNA or genes than the body has are in these bacteria. And they're a factory of hormones, chemicals that modulate and really regulate health and disease in people. And if you look at the list of disorders associated with abnormal microbiota of the gut, it goes from C. diff on one end to obesity on the other, and all the way through diseases that we all know about. Parkinson's disease, multiple sclerosis, Crohn's disease, asthma. There are just an incredible number of diseases associated with abnormal microbiota. And we're developing our tools to treat a number of diseases with C. diff because it's pure dysbiosis or pure abnormality of the microbiota. We can fine-tune the products that we need to reverse the microbiota and make it healthy. And so we're using C. diff in that regard. We can treat it, we can cure it, but while we're doing that, we're developing tools that will move into these other diseases.
Okay, so let's go ahead and turn to your study. And essentially you had a number of patients with recurrent C. diff, something you've talked about, and you treated them with either fresh, frozen, or freeze-dried fecal microbiota transplantation product via colonoscopy. So can you talk about your study?
Yes, and when we talk about fecal microbiota transplantation, what we're talking about is taking the bacteria, extracting them from a stool of a very, very healthy donor. They have to be screened very carefully. And we filter that stool, we remove the bacteria from it through 2 filtrations. And then we can give it as fresh product. We just isolated it 2 hours ago from a donor, and we put that in the colon of a patient, or we can freeze it for months at minus 80 degrees centigrade. So that means if you need it, you can just thaw it out of the freezer and give it to a patient. Now, the reason why freeze drying is important is if we show freeze drying works, just like instant coffee, we can then put it in capsules and we can give it orally to people. And it, like frozen product, can be stored for months at a time. You don't have to have a donor right there on site to treat the patient. So what our study is, we wanted to see whether fresh was more effective or equally effective with material that we'd isolated, extracted from donor stools and frozen at minus 80 degrees centigrade, very, very cold temperatures, or whether it was freeze-dried and made into powder, and we reconstituted it some months later and gave it. And what we found is that the fresh product was a little better than the other 2, but all 3 products worked very, very well and successful in this trial. But we found the fresh was the best. The lyophilized freeze-dried was the least good of the 3, and the frozen was somewhere in between. Well, when you look at the worst group, the freeze-dried, it was good enough for prime time. I mean, over 70% of the patients that were treated were cured from their disease. So it's not quite as good as fresh. So we knew we had to do something to it to make it more active. So we've added more microbiota to it now. We've put it into capsules, and that's where we're going with it. And right now in Houston, our primary way to reverse the microbiota in people with illness is with capsules. And it was this study published that you cited that allowed us, gave us the knowledge that we could move forward with these freeze-dried product.
Now, the goal to move forward and with the freeze-dried FMT product, put it in a pill form, has to be fantastic news for providers and patients alike.
Absolutely. And we're now moving, we have INDs from the U.S. Food and Drug Administration for 4 indications. One is C-diff, which we've been working on for 3 years. The other is Parkinson's disease. A third is irritable bowel syndrome. And a fourth is fatty liver disease, which is an epidemic disease in the United States associated with obesity.
Well, this is fantastic. I really enjoyed the study. And what's the next step, Dr. DuPont?
Well, the next step is to show that the pills are as effective as giving it by colonoscopy, and we have shown that now as a result of this publication. And we're moving forward with oral capsules in these other indications, such as Parkinson's disease. And I'm telling you, within 5 years, this will be standard therapy for many of these diseases which now don't have treatment. And the other use of this is in antibiotic resistance, which costs this country $30 billion a year. If you can replace antibiotic-resistant microbiota in the gut after courses of antibiotic with a few capsules of this product and totally eliminate resistance, which I think we can do, this will be a chaser for virtually all antibiotic treatment that we give in the future. So this is going to open up new avenues of research. I call it high tech or high science, low tech research. The tech is low, it's easy, it's inexpensive, but the science manipulating these microbiota in the gut is perhaps the most important science we're doing right now in medicine.
Yeah, and one little side note from your study, I noticed the fresh stool was effective 100% of the time.
That's right. That's right.
Yeah. So I mean, it's hard to imagine why anybody would want to give vancomycin anymore.
Well, this is not to treat primary disease.
Oh, okay, gotcha. It's recurring.
To prevent recurrence.
Right. You're right. Okay. All right. Well, thank you, Dr. Herbert DuPont, for your time and expertise, and congratulations on the progress in the study.
Great. Thanks so much.
You bet. Thank you, sir.
Bye-bye.